Assalamualaikum and a very good morning. Today, I will present my comparative study on ellagic acid and gallic acid in enhancing the intestinal barrier function and wound healing. And this is my contents for my presentations. Basically, we are focusing on the introduction, the research gap, objectives, the antioxidant, the potential of the ellagic acid and the gallic acids, the barrier restoration, the cellular repair, transcriptomic insights, and comparison the cell responses, and also what are the recommendations for my projects, and also the conclusion and the references. Okay, to begin, let's look at the clinical context here. The inflammatory bowel disease, which includes the Crohn's disease and the ulcerative colitis disease, is rising globally. In early industrialized countries, expected within the next decades, more than 1% of population is expected to live with IBD. Okay, Malaysia, like many Asian nations, is experiencing a steady increase in incidence, which is they can reflect our changing of the lifestyle, the urbanization, and the dietary patterns. Other than that, we have this also the colorectal cancer, which is this cancer are the third most cancer in worldwide, second leading cause of the cancer death also. And it's a major concern because yes, because it's the third most common cancer. And in 2025, IRC estimated 1.9 million new cases and over 900,000 deaths annually. Alarming uh, incident is rising among people that is younger than 50 years old, which this can be highlighting the urgent need for the preventive and the therapeutic strategies. Okay, the intestinal epithelial function or work as a dynamic barrier which is regulating the nutrient absorption, immune defense, and also the protections against the microbial invasions. And for the tight junction proteins such as zonula occludin 1, occludin, claudins are essential for maintaining the epithelial integrity. However, when the inflammatory stimuli like lipopolysaccharides or also known as LPS disrupt this barrier, the intestinal permeability will be increased, which is this leading to the bacterial translocation, oxidative stress, and also the chronic inflammation. Okay, this matter will contribute to the gastrointestinal disease such as IBD, which we already know, which is the ulcerative colitis, and also the Crohn's disease, and the colorectal cancer. As we can see here, the research gap for this research, the therapies for the IBD are quite limited nowadays. Okay, and even they offered some treatments, they always have the side effects to the patients as it comes also with the high cost. So to overcome this problem, we may create one of the affordable methods that exist to restore the epithelial barrier functions. So as shown, polyphenols from plants, particularly ellagic acid and gallic acid, have shown promise as natural alternatives due to their antioxidant and anti-inflammatory properties. So for my projects, there are 5 objectives for my research study, which is first determining the cytotoxicity between two models, which is the FHS 74INT and also the CACO2 cells. Second, profiling the transcriptomic changes through the RNA sequence analysis, and also quantifying the antioxidant activity using DPPH and FRAP assays, and also investigating the tight junction genes and protein expression through qPCR and the Western blot stain. And last but not least, assessing the regenerating capacity through wound healing assay. For the next slide, we are focusing about the antioxidant potential. EA and GA are expected to demonstrate strong activity in the antioxidant. And in the DPH assay, low IC90 values will indicate the robust radical scavenging potential. And these two compounds, if we do undergo the FRAP assay, it will show the high ferric reducing power expressed at the Strollox equivalence. And these findings suggest that both compounds can effectively neutralize the reactive oxygen species and also can balancing the redox, cellular redox, which this is critical for maintaining the tight junction integrity. Next, we are focusing on the barrier restorations. Under the inflammatory stress, the tight junction proteins such as zonula occludin 1, occludin, and the claudin 1 are disrupted by the lipopolysaccharides, or also known as the LPS, which is this leading to increase the intestinal permeability. The treatment with the ellagic acid and the gallic acids is expected to upregulate these junctional proteins to reversing the LPS induced damage. Okay, in this study, the CACO2 serve as the model. It is because the properties of the CACO2 is more better in making them ideal for the assessing the barrier integrity. And how do we want to validate it? Is like more, is it like valid or not? Okay, the validation will be performed using the Western blotting and the R2-qPCR, confirming that the polyphenols is actively restored or not. So this validations will be show the polyphenols can restore the epithelial barrier functions, and this demonstrates these both compounds not just work as an antioxidants property but also as the barrier protective agents, which can strengthening the gut integrity also. Okay, for the next slide, we are focusing about the cellular repair. So this slide highlights the dual protective roles of ellagic acid and the gallic acid in intestinal epithelial cells. So from the studies, EA and GA promote faster wound closure in treated cells. So on the left side, we see the scratch wound healing assay. The treated cell by the polyphenol cells show faster migration and repair, closing the wound more efficiently. Okay, this reflects their role in epithelial regenerations. On the right, we're focusing on the barrier restoration in the CACO2 cells. So basically, these two phenolic compounds upregulate the tight junction proteins like zonula occludins, occludin 1, and the claudins. So this can reverse the LPS-induced damage and restoring the barrier integrity at the same time. So together, these findings that ellagic acid and the gallic acid support the gut health by enhancing the both structural repair and functional protection. Okay, to understand how the ellagic acid and the gallic acid exert their protective effects, the RNA sequence analysis are performed from the study, and the results are expected to show the modulation of key pathways, including the oxidative stress response, the inflammatory signaling such as natural factor kappa B and MAPK, also known as a mitogen activated protein kinase, and last but not least is the pathway is epithelial repair mechanism. Okay, these transcriptomic changes suggest that EA and EA not act only as antioxidants, okay, but also actively support their barrier restoration and the wound healing also. And how do we want to validate these findings? The usage of the RT-qPCR and the Western blotting will be used to confirm this expression of selected genes and proteins. Okay, this approach provides the mechanistic clarity, linking the polyphenol treatment to functional outcomes in the gut health. And the RNA sequence analysis will reveal the modulations of pathway involved in oxidative stress response and the inflammatory signaling and also the epithelial repair mechanism. Okay, the pathways such as natural factor kappa B and MAPK are likely to be differentially regulated, and the validation through the RT-qPCR and the Western blotting will confirm the relevance of the selected chromatomic markers and providing also the mechanistic clarity on how the EA and GA exert the protective effects. Okay, so for the comparative cell line response in these slides, also we use two complementary cell models to capture the different aspects of intestinal health. So as you can see here, the cell analysis is we use the FHS74A cells and the CACO2 cells. And these two models are used in the different aspects. One is for the regenerations and another one is for the barrier restorations. So on the left, the FHS74A cells emphasize the regenerative mechanisms, reflecting the development of the biology. And they are highlighting the process like migration and repair, which are crucial for the wound healing. On the right, the CACO2 cells is more on the demonstrate the barrier restorations and their mature junctional properties makes them ideal for the studying tight junction proteins such as zonular occludin 1, occludin, and the claudin 1. And by combining these two models, we can see that both can capture the structural repair and the functional barrier integrity, and this can be adding to our study. This dual cell approach strengthens the relevance of the EA and the GA as a protective agents for the gut health. Okay, we have come to our the recommendations and the conclusions for the last slides. And for the recommendations, we should explore the translational application of ellagic acid and the gallic acids, which we already know that these two compounds promises as a nutraceuticals or functional food components for managing the gut barrier dysfunctions. Future research should investigate more, especially in the dietary supplementation strategies, formulation stability, and also the clinical safety profiles. And also, the clinical trials could then assess the patient's outcomes, positioning these compounds as affordable and accessible alternative to conventional therapies. And secondly, broaden the therapeutic scope beyond the IBD. Okay, the potential of ellagic acid and gallic acid should be explored in related gut disorder such as colorectal cancer and the metabolic disease. And also, the comparative studies across different polyphenols may reveal synergistic effects, which is this can be open new avenues for combination therapies. Okay, the last but not least, the conclusion for these studies. Okay, this research highlights the potential of the ellagic acid to restore the intestinal epithelial integrity under the inflammatory stress. Their dual roles as antioxidants and the barrier restorative agents makes them promising candidates for managing the IBD and the related gut disorder. And by using these two cell models, which is the FHS74INT and the CACO2 cells, have been captured that cell type specific responses, which is we are focusing on one cell model in one responses. So this can enhancing more our research. Okay, so from the regeneration to tight junction restoration and adding will be adding more in the translational depth to our findings. Overall, this supports the development of safe, affordable therapies that target multiple aspects of gut health. So, these are some of my references for my studies, and thank you for listening for my presentation for today. Thank you.